Good morning and good afternoon, everybody. Thank you again for attending the webinar this morning. We're going to go ahead and get started. My name is Abi Saharia, and I'll be the host here today for our webinar titled Building Relevant Cell-Based Assays for Co-Stimulatory and Co-Inhibitory Checkpoint Receptors. Before we get started, a couple of administrative announcements. This webinar is being recorded, and a copy of the recorded webinar as well as a copy of the slides will be available to you shortly after the end of the webinar. In addition, if you have any questions during the course of the webinar, please type those into the Q&A box or the chat box that you can find on the drop-down menu of the WebEx program. Before I introduce Dr. Jane Lemerdin, who is going to be giving us presenting most of the webinar on our cell-based assays for checkpoint, co-stimulatory and co-inhibitory receptors, I would like to take a couple of minutes to introduce those of you who might be new to DiscoverX to the offerings that we have at DiscoverX, particularly to drive the development of immuno-oncology drugs based both on target-based as well as phenotypic platforms. At DiscoverX, we are a leading provider of cell-based assays focused on driving or enabling the drug discovery community, particularly within the cancer immunotherapy drugs, driving it all the way from screening to the clinic. The target-based assays that we have are largely dependent on our enzyme fragment complementation that Dr. Lemerdin will talk about in greater detail, with applications for these assays for screening, lead optimization, development of bioassays for potency and stability testing, as well as further downstream, looking at neutralizing antibody assays for immunogenicity testing in late-stage clinical trials. In addition to our target-based assay platform, we also have a phenotypic assay profiling platform, which is the Biomap Human Primary Cell-Based Assays. And these primary human cell-based assays have a tremendous number of applications, in particular looking at efficacy and biomarker selection, preclinical safety studies, as well as combination studies, all of which together enable the clients to go all the way from screening to the clinic with the use of the DiscoverX assay portfolio. So in particular, as we talk about the use of the checkpoint assays for screening and lead optimization, this can enable the development of molecules, identification of lead molecules with this. And then as the molecule moves further downstream into development, these same assays can then be used for lot release testing for molecules that you would have. In addition to that, the services offered by Biomap, as I mentioned earlier, enable the molecules, the most appropriate molecules to be selected based on efficacy and safety studies during preclinical testing, and then also look at combinations to identify the most appropriate combination to take forward into the clinic. So altogether, that again provides a very comprehensive portfolio of assay, solutions for immuno-oncology drug development, with the focus of today's webinar really being on the checkpoint assays that can be utilized for screening all the way through development of lot release assays using these molecules. So with that very brief introduction, I'd like to now turn this over to Dr. Jane Lemerdin, who is going to be presenting the webinar on the development of these, or the building of relevant cell-based assays for co-stimulatory and co-inhibitory checkpoint receptors. Dr. Lemerdin is the director of our R&D team here at DiscoverX, and she has been instrumental in the development of all of these assays and pioneered the use of our enzyme fragment complementation technology for the development of all of the checkpoint receptor assays that we'll be talking about today. So without further ado, I'll turn this over to Dr. Lemerdin. We are all very excited and looking forward to the webinar today.
Thank you, Abi, and thank you all for taking time out of your busy day to join us for this webinar. So I'd like to start and just by referencing how much excitement there is in the field of cancer immunotherapy. And obviously this is the interest around building checkpoint assays, is to build new therapeutics for cancer immunotherapy. Now, as you're all aware, this was a field that was designated the Scientific Breakthrough of the Year back in 2013 by Science Magazine because of all of the great promise that this field holds. And the reason this is so exciting is it's an approach that's currently being used in oncology that is quite different from the past, in that it harnesses the immune system of the patient to battle tumors. And we're able to do so by selectively activating T cells in several different ways. So utilizing the ability to modulate different types of receptors that are on the surface of T cells. There are two different classes, as you can see in this cartoon on the right. You have receptors that are involved in stimulating the immune system or T cell activation. And so drugs can be developed to stimulate those particular receptors. And that's referred to as pressing the gas pedal on the immune system. And then conversely, there are a number of receptors, such as those shown here in this box, that represent inhibitory receptors in affecting inhibition of the immune system. And so if you block those receptors, you can release the brakes on the immune system. So this field really took off based on the clinical success of molecules targeting two of these specific inhibitory receptors, CTLA4 and PD-1. And since that time, there has been an immense amount of work in showing that not only in monotherapy, but now in combination therapy with other small molecules and other antibodies, that you can improve the outcome for the patient by combining these checkpoint receptor molecules, and thereby customizing the treatment depending on the patient's tumor type. So as I mentioned, the T cell stimulation can occur through two different types of receptors. You can have either co-stimulatory receptors or co-inhibitory checkpoint receptors. And there has been an expansion of our knowledge over the years as to the types of receptors that are involved, and that continues to grow. And as those types of receptors are being identified, what we realize is that we need more and more tools that are specific and allow us to develop specific molecules and develop new therapeutics. So again, this is just a cartoon elaborating on the concept of the gas pedal versus the brake, where on the left we have activating receptors, where the therapeutic modality would be to stimulate those receptors with agonistic antibodies, or in some cases, multimerized ligands. And then on the other side of the coin, if you will, we have the inhibitory receptors, and the number of those, there's only a subset really that are known that are shown here, but these are all common targets that a lot of groups are interested in inhibiting. And again, the therapeutic modality there would be blocking antibodies that are specific to these individual receptors. So the challenges with developing assays for checkpoint receptors are many, and primarily because of the difficulty of working with primary human cells. As we all know, it can be very difficult to develop these assays to identify the appropriate cell types and an appropriate readout. It does require people with advanced skill to handle these cell types, and while that is something that can be accommodated in the early stages of drug development, typically on the back end when you have assays that need to go into a QC environment, working with primary cells or more finicky cell types can be very challenging. And if you add on top of that the amount of variability that one sees due to working with primary cells, and also the donor heterogeneity, so lot to lot variability, then that makes the assays that much more challenging to work with in terms of robustness and reproducibility of the outcomes. The co-cultures often required for these types of assays are also long and complicated protocols, which don't translate well for a QC environment. And then probably the most important is that a lot of these types of assays are not specific for the target receptor. And so when you're looking at inhibition, you can't be certain that from IL-2 secretion, for example, that you're actually inhibiting the receptor that you're targeting. So there's a great need in the field for assays that have that level of specificity. So when we started thinking about designing assays for checkpoint receptors, the first one that we decided to take a look at was PD-1 because there's so much information about how this receptor signals. And we are most interested in looking at very proximal events to receptor activation. So in looking at the signaling scheme that is known for PD-1, as shown here on the right, typically what is known is that PD-1 itself is a receptor that within its cytoplasmic tail harbors some inhibitory motifs known as ITIM or ITSM or switch motifs that become phosphorylated when the receptor engages its ligand. And here the ligand is being presented on the surface of a tumor cell or antigen-presenting cell. So when this event occurs, what happens is you have signaling kinases, Stark family kinases to be specific, that phosphorylate these motifs. And that results in recruitment of SH2 domain-containing proteins such as the SHIP phosphatases. And that dephosphorylation event then results in the silencing of signaling kinases that are acting at the T-cell receptor and attenuation of T-cell receptor activation. So that's what occurs in vivo. And what we wanted to do was to build an assay that looks at this very early signaling event of SHIP recruitment to the cytoplasmic tail of PD-1. So the way that we went about this was to use our validated enzyme fragment complementation technology, where we're using a split beta-galactosidase enzyme. So for those of you not familiar with our system, we have a fragmented enzyme where the beta-gal N-terminus, or we call enzyme donor, is a 42 amino acid unstructured peptide, and the remainder of the enzyme, which we call enzyme acceptor, is about 110 kilodaltons. So when these two proteins are separated, they have very minimal enzymatic activity. And so we use this system to actually look at the interaction of two different proteins that are brought together by their own biology. And when they do so, they're able to bring the two fragments into proximity to allow complementation and reconstitution of an active enzyme. And we can detect this state by adding enzyme substrate and then measuring out a chemiluminescent signal, which can be read on a plate reader. So the way that we approached building a PD-1 signaling assay is shown in this cartoon here on the left. So our T-cell model is GERCAT cells, where we've engineered in the full-length PD-1 receptor, and at the C-terminus of the receptor, we have our little PK tag. Engineered into the same cells are a SHIP protein, which we've fused to the enzyme acceptor. So in the absence of any ligand binding, there's no signal or very low signal coming from these GERCAT cells. But when you co-culture them with our tumor model, if you will, which is U2OS cells that are expressing either PD-L1 or PD-L2, what you see is the ligand engagement interacting with PD-1 recruits the SHIP phosphatase, and that results in enzyme complementation, and we get a signal when we add our substrate. The nice thing about this assay is it's very straightforward. It's a homogeneous readout. So what we're able to do here, and you can see from the workflow, is that we plate our GERCAT PD-1 expressing cells, and we add our anti-PD-1 antibody. We incubate that for an hour at 37, and then we add our U2OS or PD-L1 or PD-L2 presenting cells, or our ligand presenting cells. And then we incubate those for another hour to 2 hours. And then we add our detection reagents, incubate for another hour, and read that on a benchtop luminometer. So all of this process takes less than 5 hours to detect a signal. So this slide shows you an example of a response to ligand presented on the surface of a UTOS cell. Here we're using PDL2, and you can see here the blue dots here correspond to the native UTOS, which express very little to no PDL2 ligand. But when you titrate in increasing amounts of cells that are presenting PDL2, you see a corresponding increase in the assay window in response to ligand. And you can see that at the highest point, you can get a 70-fold window, but you probably wouldn't want to use that many cells. So now we see that if we stimulate the assay with PDL1 or PDL2 and then incubate with an anti-PD1 antibody, we see very robust inhibition of both PDL1 mediated or PDL2 mediated signaling. And in both cases, the IC50 is very similar for the same antibody. And we know from the receptor density that the amount of ligand on the ligand-presenting cells is very comparable. And these result in assay windows of around 25 to 30-fold. So it's a very robust assay. The assay is also compatible with anti-ligand antibodies. So in this case, we're showing you both on the same graph, either anti-PD1 or anti-PDL1. And again, the rank order in this particular case, these are commercially available anti-PD1 and anti-PDL1 antibodies. And we always see the same rank order in this assay. So this assay is suitable not only for biologics, as many of the data that I've shown you so far would reflect, but also for small molecule inhibitors. So the data on the left are showing you inhibition with the therapeutic molecules, Keytruda and Opdivo. And you can see that the performance in this assay for both of these molecules is very similar. And again, the windows are very similar to what we saw on the previous slides. But interestingly, on the right, if you'll recall how this assay works, where we're looking at a phosphorylation event that results in ship recruitment, that we're able to use promiscuous Src kinase family inhibitors to show that you can inhibit those particular types of molecules in this assay. We also know from very recent data that an inhibitor of the interaction between PD1 and PDL1 is also able to produce an inhibitory response in this assay. So it should be suitable for looking at ligand receptor interacting blockers as well as kinase or other intracellular signaling molecules involved in signaling. So this is another example of the specificity of the assay. In this case, we're showing you data from the anti-PD1 antibody, which is shown in dark blue, and an antibody targeting another checkpoint receptor, the TIM3 receptor, is not active in this assay, or only has very marginal activity. So we can see again that the response is highly specific to the PD1 receptor and its ligand. Importantly, what we see is that the assay is very reproducible. This is data showing you 3 experimental replicates of an anti-PD1 antibody prepared on the same day and run on the same plate. So within a single experiment, you see the very tight signal to background and IC50 values with an RSD of less than 4%. So we have compared this assay to one of the reporter gene assays that's on the market, and we're showing you that contrasting data here. And the important thing to note is that the same antibody, which is a commercial anti-PD1 antibody, was used for this comparison. And what you can see, that's the PathHunter Assay, as it's called here for PD1, gives you a very low IC50 for this PD1 antibody relative to the gene reporter assay. So it was a 54 nanogram per mil versus 927 nanograms per mil. Importantly, the assay time is also much shorter, less than 5 hours to run our assay versus 22 hours for the gene reporter assay. And the assay window is also more robust with this particular antibody. So we've now extended our portfolio to include another inhibitory receptor, which is the CTLA4 receptor. And importantly, we're able to show that based on the literature, that the shift recruitment concept is also applicable to CTLA4, as you can see here. So we've built an assay very similar to what we've done for PD1 using CTLA4. And so in this particular case, CTLA4 responds to two different ligands, CD86 and CD80. I'm showing you data here for CD86. And this is a ligand-presented cell on the surface of U2OS. And you can see a nice dose-dependent response to CD86, while if the cell line is co-cultured with PD-L1 presenting cells, you see no response, so demonstrating the specificity for the ligand. Similarly, if we look at the specificity of response to antibodies, that in this particular assay, again, was stimulated with CD86 and then incubated with either Ipilimumab or Keytruda, we see the expected inhibition profile for Ipilimumab, but not for Keytruda. So again, the assay is very specific. And again, if we compare this assay to a gene reporter assay for the same target, again, we show that the PathHunter assay is more sensitive in that the IC50 is only 123 nanograms per mil versus 1.68 micrograms per mil. The assay time is also still shorter. The kinetics are slightly different for this assay relative to our PD1 assay, but still can be run in a single 8-hour day. And the assay window is fourfold here, slightly lower than the gene reporter assay. But again, robust enough and very good sensitivity compared to the other reporter gene assay. So to summarize these two co-inhibitory receptors, the benefits of these particular cell models, we are looking at receptor activation through co-culture. So we are essentially building a model that's very similar to what one would use if you were using primary cells, but with the benefit of not needing to use primary cells because we've engineered biologically relevant responses into stable cell lines. The protocols are very fast, one of them taking less than 5 hours, the other less than 8 hours, and they're highly sensitive with 10 to 15 times more sensitivity than the other gene reporter assay. And importantly, these can be applied not only to screening for biologics, but also small molecules. And we know a number of groups that are using this not only for looking for antagonists, but also looking at agonists for autoimmune. So if we move on to the other category now, to talk a little bit about the portfolio or the approach that we've taken for our co-stimulatory checkpoint receptor portfolio. And what many of you may know is that a large number of the activating receptors on the T cell fall into a family of, a gene family, if you will, of TNFR superfamily members. And the nice thing about that is that they will all signal through canonical and non-canonical NF-kappa B pathways, providing us an access point for building out an assay. So although the canonical pathway is something that has been well studied in a number of receptors, it's also known that many of the important co-stimulatory receptors, such as 4-1BB and OX40 and GITR, also signal through the non-canonical NF-kappa B pathway, and that pathway is shown here on the left. So as we tend to build our assays, we like to identify a reporter that is as proximal to receptor activation as possible to eliminate the possibility that other pathways could be impinging on the readout. So when we looked at the non-canonical pathway, one of the readouts that we were interested in was this protein NIK. So NIK is the NF-kappa B inducing kinase that is maintained at very low levels in the cell ordinarily. So in the absence of pathway activation, this protein is actively degraded by the proteasome. However, when the pathway becomes activated by ligand engagement, the NIK protein is able to accumulate because the proteins that regulate it are recruited away. And this stabilization event can be measured by labeling this protein with our EPL tag. So the consequence of NIK stabilization is that you have activation of P100. It becomes processed and interacts to P52 and then interacts with RelB and enters the nucleus. But what we wanted to do was to look at something that was occurring a little further upstream, so we're focusing on NIK stabilization as our readout for a number of these assays. I'll just point out parenthetically that we also have built a number of assays using the canonical NF-kappa B signaling pathway by looking at I-kappa B degradation, but I won't be talking about any of those assays today. So this is a cartoon that describes how we build out our NIK stabilization assay. It's very straightforward. Essentially what we do is we take a full-length NIK protein and put our small EPL tag on the C-terminus. So this gives us the most sensitive ability to modulate, to detect modulation, of the protein. And we can introduce this NIK EPL fusion into any cell type of interest. What we've chosen to do here is to show you this in U2OS, but it can be introduced into other cell types that may endogenously express the receptor of interest. So once the protein is expressed in the cell type of interest, you then plate them, stimulate them with your ligand, and then when your time course is done, you add detection reagent and substrate and read that on a plate reader. So very few manipulations are required. So some examples of this assay format that have been used to look at endogenous TNFR superfamily receptors in these U2OS cells are shown here on this slide. So we have one example here where the tweak receptor is activated by addition of tweak ligand. And here the HVEM receptor, which is another co-stimulatory receptor, is activated by its ligand light. In both cases, the EC50s are in the low nanogram per mil range, and the assay windows are on the order of 3-fold or better. Now we've applied the same approach to looking at CD40, which is a very important target for immuno-oncology and also other immune modulation. And what you can see is that NIK is a relevant readout for this particular pathway. And so what I'm going to show you are some data that we've developed for CD40, looking at multimerization of ligand and monoclonal antibody data. In this particular case, the response to CD40 ligand results in an assay window of around 3-fold, but it is a reproducible 3-fold. The assay time is 4 hours, and this is again using our current NIK cell line in U2OS cells that endogenously express CD40. So it's long been known in the literature that soluble CD40 ligand is in vivo not able to produce the optimal activation of the NF-kappa B pathway or downstream signaling in general. And so what we show here is that you can see a response to soluble CD40 ligand. But when you compare that to what's observed when you enforce trimerization of the receptor, which is known to produce increased signaling activity in vivo, is that we can see a much improved efficacy and potency of the ligand if we oligomerize that by oligomerizing it with an anti-HIF antibody, where the CD40 soluble ligand has a HIF tag on it, obviously. So this is, again, recapitulating the biology that you would expect by presenting the ligand in a way that is more efficacious at activating the receptor. And then similarly, it's well known that agonistic anti-CD40 antibodies in vivo are able to cluster and activate the receptors, but that activity is dependent on Fc gamma receptors. So while we did not use any cells that have Fc gamma receptors, you can see a response with agonistic antibodies that are produced or presented as soluble, if you will, antibody. So the assay will respond to all three, and if you were to cross-link the antibodies, you would have a stronger response than is shown here. So we've also approached building the OX40 assay. This, again, is shown in the literature to be dependent on NIK for signaling through the non-canonical pathway. In this particular case, we are introducing the OX40 receptor into our U2OS NIK background. So this is engineered to be specific for OX40 ligand. And again, see an assay window of 4-fold and assay time of 5 hours, which is slightly longer than is required for CD40. And I'll just point out that the assay is extremely robust. The data I'm showing you here is for 96-well format. We find that the assay window is actually improved if you're working in a smaller well volume, such as 384 well, which may be more conducive to screening a larger number of antibodies. And finally, for the co-stimulatory receptors, we've also built an assay now for CD137, or also known as 41BB. And again, NIK is a central component of the signaling pathway here downstream of CD137. So as you can see here on the right, the response to soluble ligand produces about a 3-fold window with an assay time of 6 hours for this particular ligand. And we've also validated this assay with both oligomerized ligand and an agonist antibody, in this case Urelumab. So this slide is just showing you some of the data that we've obtained looking at different ligand sources. And this is something we've seen with a number, and I'm sure many of you are familiar with this, that different providers of ligand can have different qualities that can affect your assay. In this particular assay, we've looked at 3 different vendors. Vendor A produces a soluble ligand that has superior performance in our assay. When we tested another vendor that produced an Fc conjugated ligand, the response was suboptimal for us. It only produced about a 1.7-fold window. And we looked at another vendor where the molecule was his tagged, and we both ran it as a soluble and as an oligomerized molecule, and the performance was very similar in this particular assay. We've noted that there can be some changes in response to different serum concentrations that might change that rank order slightly, but in general, the soluble ligand that we're using and that we recommend with our assay gives the best performance in this format. So finally, we've also tested this particular assay with an agonistic antibody and compared that to the response with the same soluble ligand. And what you can see is that the response to Urelumab is superior to the soluble ligand, as you might expect. And so the assay is suitable for testing both soluble ligand and antibodies. So to summarize this platform, if you will, for our co-stimulatory receptors. Again, what we're looking at is measuring activation of these checkpoint receptors. That's the intent of the ability to detect agonistic antibodies or multimerized ligands that could be used as therapeutics. Again, we've engineered a system in stable cell lines that don't require human primary tissues, but still give you a response that is biologically relevant. The protocol is, again, quicker than some assays in that we require less than 6 hours typically for the readout of the assay. I think it's important to note that due to the ease of running this particular assay, that it's really suitable for supporting not only the drug development phase, and these assays are suitable for running in 96, but also in 384. But the same assay can progress and transition into use in QC lot release because they are very robust readouts. Finally, although we are working on additional assays, building other assays in this family, this particular approach is customizable to other receptors. So if you don't see the receptor that you're interested in, but would like to use this particular approach, then we can actually build those assays for you as a custom assay. And it can either be using the canonical or the non-canonical NF-kappa B signaling path. So I'd like to just summarize now the available checkpoint assays that we have as of June. We definitely have a number of other ones in development. As you can see here on the left cartoons, there are a number of assays that we have stars here. Those indicate assays that are currently available. And there are others on this particular schematic that we are actively working on as well. So we encourage you to reach out to us if there's interest on that part. And I'll also put in a plug for our other assays that could be of value to you. We have over 40 interleukin receptor assays that include our GM-CSF and MCSF assays that are, again, have some utility in the IOX space. So I would just like to summarize. I hope what I've shown you today is that we have a number of reporter assays that have been engineered into stable cell lines that have the biological relevance but overcome the limitations of primary cell-based assays. They are biologically relevant in terms of the measurement of activity of the receptor. The assays are all based on the native receptor biology and have high specificity of signal. We are able to show that they have a consistent and reliable response, which makes them very useful not only in early screening, but later on in the drug development process for QC lot release assays, both because of their simple protocol and their high reproducibility. And I'd like to also point out that the assays now, as they're currently provided, they are stable cell lines for continuous culture in the lab. However, we have shown in the past that these are adaptable into frozen ready-to-assay format, and this could be something that your own in-house groups can do, or we can do as a custom development for you. So with that, I'd like to close and thank you again for your attention. If you have any questions, please feel free to enter them now.
Thanks, Jane, for a very, very interesting and informative webinar. There's a number of questions that are already starting to come in. Again, if you have any questions, please type them into the Q&A box or the chat box so that we can get those addressed right away. So a couple of questions that have come in already is with regard to the PD-1 and the PD-L1 assay. And the question is, can the PD-1 assay be used with cells expressing endogenous PD-L1?
So in principle, yes. I think one thing that it's important to be careful about is that different cell lines, especially cancer cell lines, have some endogenous beta-gal activity in them. So what we find is we have to be very careful about the titration and identifying a cell line that has very low beta-gal activity that will not contribute to background in the assay. So we definitely can use other cell types, but what we've elected to do for engineering an assay that has broad applicability is to use a cell line such as U2OS that had a lower beta-gal background.
Thanks, Jane. So another question that's come in is, can these assays be used in the presence of serum?
Which type of serum?
It says human serum.
Oh, human serum. Yes. So we haven't directly tested that with the PD-1 checkpoint assays, but we have done this for the NIK assays. If you are using them for an immunogenicity type of assay where your antibodies are going to be diluted. We have tested many different assays up to this point and shown that up to typically 10% normal human serum is well tolerated by the assay.
Great. A question that's come in is also regarding the co-culture of these assays. So in particular, does co-culture of assays affect the reproducibility of the assay?
So what I think you mean perhaps between experiments.
Yep.
So what we've seen is that the IC50s that we obtain are still within a very tight window. So it's not dramatically different from what we see for other assay formats. So I would say between 3- and 5-fold difference between experiments. If we move to the bioassay format, we're trying to optimize those so that they will have a tighter window.
Great. One of the last questions that we could address here with regard to time is question regarding the co-stimulatory checkpoint assays. Have we tested any of those assays in a co-culture based approach?
So because we were originally developing these for to detect agonistic antibodies, we didn't take that as our first approach, although we have been looking at that for our GITR optimization. So we definitely have an interest in doing that for the therapeutic areas that are looking at antagonist molecules. But we haven't yet done that testing because up to this point we really haven't needed to.
All right, thank you again for attending the webinar this morning and for the stimulating questions that we've received. If there are any questions that we've not been able to address, please type those, send those via email to the email addresses shown here. Again, to reiterate, if there's any questions around assays that we haven't talked about but you have interest in, you can send them directly into the custom assay development team or discuss that with your account managers, and then we can get that initiated via discussion with the R&D team and Jane's team in particular. In addition, if there's any technical questions that you have with regard to any of the material presented during the webinar, please feel free to reach out to the tech support team at in North America at support.us at DiscoverX.com, or in Europe at support.Europe at DiscoverX.com, with countries from the rest of the world also sending their questions to support.us at DiscoverX.com. Additional information about the assays is available at DiscoverX.com/checkpoint. And with that, I'd like to thank you again for your time and wish you a good rest of the day and a great long weekend. All right, have a good one. Thanks, bye-bye.